Hello, everyone. So let's continue our discussion on the molecular nature of the genetic material, more specifically, a DNA molecule. And even more specifically, the DNA molecule that was described by Watson and Crick in their important paper published in 1953. So what we've done is, we described the molecular model that they proposed. And what we want to now do with that understanding is relate structure to function. So one of the first functions we're going to be describing is replication itself. So the ability of the genetic material to faithfully and accurately replicate itself, which of course would be important prior to any cell division. So we're going to begin to explore how, again, the structure of the molecule, as described by Watson and Crick, is able to fulfill or achieve this specific function. So this is going to draw from material out of chapter 11 in your book, and here are the pages I would ask you to look at for more detailed explanations of some of the things we'll cover. There's a couple of problems at the end of the chapter that I would suggest you look at as well. So as I said last time, Watson and Crick concluded their publication or their paper with a very brief statement suggesting that, again, because of the characteristics of their model, specifically the nature of the base pairing, very specific, two types, T and A and G and C, is that they suggested that that particular nature of the base pairing that exists within the molecule suggests a copying mechanism. So what did they actually mean by that? What they were describing is what we would nowadays call a semi-conservative replication. So the idea would be is that in this diagram here at the top, you have a double-stranded molecule of DNA, two single strands held together by the hydrogen bonds of specific types of base pairs, G, C, and A and T. And so what they were suggesting, and also in an anti-parallel configuration, and then what they were suggesting is that all you would have to do is separate those two strands, convert them from a double-stranded form to a single-stranded form, as shown here, and then use these single strands as templates for new DNA synthesis in order to create two double-stranded daughter molecules. So the idea would be is that for each of these individual single strands of DNA, they would serve as templates for new DNA synthesis. And how would those new strands of DNA be created? Well, it'd be created based on the base pairing rules. So G with C, or C with G, G with C, T with A with T, and so on. So again, if you compare the sequences of base pairs in these two daughter molecules relative to each other, as well as relative to the original molecule, because of the specificity of the base pairing, all three of those molecules are exactly identical to each other. So this is what Watson and Crick were suggesting, and it seems to, it's so simple and straightforward that you would think, well, it has to be correct. Okay, so you can't just assume that, you have to determine that experimentally. So individual laboratories set out to try to explore experimental designs that could prove definitively that DNA replicates in a semi-conservative manner. So as part of that process of experimental design is that, you can come up with other alternative mechanisms for replication. So they're described here. This is the conservative model and the dispersive model. So this is what I've already described, semi-conservative, as suggested by Watson and Crick. So you have two original strands of DNA in this parental molecule of double-stranded DNA. They're pink. So that's old DNA. So you separate the two single strands, use them as templates for new DNA synthesis, and you're going to create two daughter molecules that are hybrids. One strand is pink, old DNA. One strand is blue, newly synthesized DNA. Okay, that's semi-conservative replication. So what's conservative? So the idea with conservative replication is that the two single strands in the original parental molecule of double-stranded DNA, they're separated. Each single strand serves as a template for new DNA synthesis. But when you put together the daughter molecules, double-stranded daughter molecules that result from this replication process, you put the two original parental strands back together, and the two newly synthesized strands are put together to create the second daughter molecule. And that's possible through, again, the base pairing rules. So that's, in other words, what they're suggesting is that if you took these two green templates here, or these newly synthesized DNA, off of the pink and the blue templates, so the green represents newly synthesized DNA, and if you look at them, when you align them in an anti-parallel configuration, they exhibit proper Watson-Crick base pairing. T with A with T, C with G, and G with C. So this conservative mechanism for DNA replication is at least a theoretical possibility based on, the base pairing rules of the Watson-Crick model. So again, you want to distinguish between those two possibilities experimentally. So they actually came up with a third theoretical alternative. This is what's called a dispersive model. So here the idea, again, pink is old DNA, blue is newly synthesized DNA. So what they said is that the two parental strands are separated, serve as templates for new DNA synthesis. But when you put the two daughter molecules together to recreate a double-stranded DNA molecule, you can see now that you're putting it together in such a way that each single strand of DNA in the double-stranded daughter molecule is part old and part newly synthesized DNA. And that's a possibility. It seems to add additional complexity to the overall process of replication, but at least it's a theoretical possibility. So they came up with three possible mechanisms for replication that they wanted to distinguish between experimentally. So how would you do that? So the idea is the following, is that if you look at these populations, so these populations of daughter molecules that are created after one round of replication or two rounds of replication, here, here, and here, and here, and here, those populations are different in terms of the composition of the DNA molecules in those populations. So it's probably easiest to see, down here, look at the second generation, after second generation of DNA molecules after two rounds of replication. So you see two of the daughter molecules, 50% of the population is newly synthesized, right? Fully newly synthesized DNA. And two of the molecules are hybrids, part old, pink ribbon, and part newly synthesized. So you compare that to the population produced after two rounds of replication if DNA replicates conservatively. In this case, that second generation, the three of the four daughter molecules are going to be fully 100% newly synthesized DNA, and one molecule, 25% of that population, are going to be old DNA, the original parental DNA, the pink DNA. So that's different from this population of molecules that's produced after two rounds of replication using semi-conservative replication. And with dispersive, again, what you see is there's four daughter molecules here, and they're comprised of, all four of them contain some amounts of old and new, newly synthesized DNA. So all three of those populations are different. So if you could come up with an experimental means by which you can distinguish between old and newly synthesized DNA, then you could distinguish between these models. So that's what they eventually came up with. So they used a differential labeling technique. Okay, and we've actually seen this before. If you remember the Hershey-Chase experiment, they used differential labeling. They made proteins radioactive, with one radioactive isotope, sulfur, and they made DNA radioactive, again, with a different radioactive isotope, phosphorus. So that's differential labeling. Protein allow you to distinguish between proteins and DNA. In this case, what you're doing is you're trying to come up with a scheme using a label that'll differentially label old and new DNA. Okay, so how do you do that? Well, what they decided to use was a density isotope of nitrogen. So not a radioactive isotope, but a density isotope of nitrogen. And so, in other words, you could make old DNA of a different density than newly synthesized DNA, and therefore distinguish between the two different types, and then be able to distinguish between these different populations of DNA that are created through 1 round or 2 rounds of DNA replication. So we've talked a little bit about this idea of how could you distinguish between different densities of a DNA molecule? Well, you can use a cesium chloride gradient. So you load your DNA into a solution of cesium chloride, you centrifuge it, and then again, the DNA will band out within that density gradient at a position equal to its own density. Okay, so the two different labels that they use are N14 nitrogen and N15. So N15 is a heavy density isotope relative to N14, the more common form of nitrogen in nature. So if you have DNA that is, and again, remember, there's plenty of nitrogen in DNA. I mean, they chose nitrogen because there's lots of it in DNA in the form of the nitrogenous bases. Okay, so what they did is, if you had fully N14 labeled DNA, that would be the low density, so that would band out at the top of your gradient. If you had fully N15 labeled DNA, that would be a DNA that would band at a higher density position within your gradient, so at the bottom of the tube. And if you had hybrid molecules of DNA that were labeled partially with N14 and N15, you get an intermediate density. It would band out somewhere in the middle of your gradient. And again, that's what you see in these different populations, either fully, say this would be N15 labeled pink, the blue would be N14 labeled, and then you have these hybrid molecules that would be labeled with both N14 and N15. Okay, so this is the way that they went. So this is actually the work of, again, another one of these classic experiments of molecular genetics performed through the 40s and 50s. And so this is the work of Meselson and Stahl. So again, a very important, classical experiment, again, because it answers a fundamental question. And the design is very simple. So again, the fundamental question is that it reveals the way by which DNA replicates. Okay, so this is what they did for the basic experimental design, is they grew a culture of E. coli, again, for these early experiments in molecular biology or molecular genetics. E. coli was the organism of choice, again, because of its biological characteristics and ease of manipulation in a laboratory and so on. So what they would do is they'd grow a culture of E. coli in a nutrient medium that contained N15 nitrogen. So as those cells were growing, they would metabolize the nutrients. The N15 would be incorporated into their DNA. So after an overnight culture, you harvest these cells, you extract the DNA, you analyze in a density gradient, and all of the DNA is going to be fully labeled with the N15 isotope. So you get a band of DNA at the bottom of the gradient. Okay, that's their time zero. That's their starting point. So then they actually performed the experiment. So then what they did is they took some of those cells, they inoculated a new culture, a culture flask containing nutrient medium that now contained the N14, the lower density isotope of nitrogen. And they allowed for 1 round of replication in the presence of N14, 2 rounds of replication in the presence of N14, and 3 and 4 and 5, they went, they did, maybe 10 to 15 different generations or rounds of replication. And what they would then do is that they would extract the DNA and they would analyze its density in these cesium chloride gradients, and they'd analyze the banding pattern. So first of all, how were they able to determine, how long it takes for the DNA to undergo one round of replication or two rounds of replication or three rounds or whatever? So again, as I said, E. coli was one of these standard organisms used in the laboratory for all kinds of studies, but certainly for, early genetic studies, molecular genetic studies. And so, its biology was very well understood, well characterized, all aspects of its biology. And so what they knew is you could, under the artificial conditions in the laboratory, is you can manipulate those conditions such that you can actually control the rate of cell division. So under optimized conditions in the laboratory, after about 20 minutes, you can get a doubling of the number of cells in the culture. So in other words, the time for the cell to divide is over a period of about 20 minutes, 20 to 30 minutes. We'll just say 20 minutes. So the DNA, before a cell can divide, the DNA has to replicate itself. So in other words, the DNA is replicating in under 20 minutes. So they would just time their experiment. They would allow the cells to grow for 20 minutes. That's one generation, one round of replication, 40 minutes, two rounds of replication, an hour, three rounds of replication, and so on. That at those time points, they would harvest the cells, extract the DNA, and analyze the DNA in the density gradients. So how do you then interpret those banding patterns to distinguish between those three models of replication? So what are the predictions, in other words? So again, here is the predicted banding pattern for semi-conservative replication. So again, we start with fully N15 labeled DNA, so it'll band out at the bottom of the gradient. After one round of replication, in the presence of the N14 isotope, you'll get two daughter molecules that are hybrids. So you predict a band in the middle of the gradient. And after two rounds of replication in the presence of the N14 isotope, you predict, you predict that two of the daughter molecules are going to be hybrids and two of them are going to be fully newly synthesized, so fully N14 labeled. So you predict two bands in your gradient, one at the hybrid position, the middle of the gradient, and one at the top of the gradient at the N14 position, the low density. So that's a very specific prediction in terms of how the DNA should band out in those cesium chloride gradients. And that pattern is different from either conservative replication or dispersive. So here's the prediction for these two alternative models. So again, remember, in both cases, our starting point is fully N15 labeled DNA, so a band at the bottom of the gradient. So if we look at conservative replication, after one round of replication in the presence of the N14 isotope, again, here you create a daughter molecule that's fully unlabeled or fully N14 labeled, and the other daughter molecule is fully N15 labeled. So you should predict two bands, one at the bottom of the gradient and one at the top of the gradient. Okay, so that's different than what we just predicted with semi-conservative replication. After two rounds of replication, using this conservative model, now three quarters of the DNA molecules present in this population are fully N14 labeled, and 25% are fully N15 labeled. So you predict two bands, again, at the bottom of the gradient, the high density, and a band at the top of the gradient, the low density. But now, here the distribution is 50-50. Here it would be 75-25. 75% of the DNA is fully N14 labeled, and 25% is fully labeled. Okay? So a different prediction from conservative replication relative to semi-conservative. So what about dispersive? So again, dispersive, after one round of replication in the presence of the N14 isotope, here's the result here. You get hybrid molecules, so you predict a band somewhere near the middle of your gradient. And after 2 rounds of replication, what you create are, using this model of replication, dispersive model, you create 4 daughter molecules, all of which contain some N14 label and some N15 label. So all 4 of those molecules are still sort of hybrid molecules, but the N15 is being diluted. There's less of it relative to N14. So even though these are all hybrid molecules in the second generation, relative to the first, or similar to the first generation, the relative amounts of N15 nitrogen in those daughter molecules of that second generation is less. Therefore, the density is going to be lower. So the prediction would be is that after 2 rounds of replication in the presence of the N14 nitrogen isotope with dispersive replication is you get a smear. You get a band that extends from the top position of the gradient, this fully N14 labeled position, and the middle of the gradient, the hybrid position. So you get a smear. Okay? So again, those predictions are different for dispersive replication relative to conservative or semi-conservative replication. So all you have to do is perform the experiment, analyze your bands, and draw a conclusion. So what did they observe? Well, this is what they observed. Starting point times 0, fully N15 labeled, 1 round of replication, the DNA banded out at that middle position. So that immediately rules out conservative replication. That's the predicted outcome if the DNA is replicating either semi-conservatively or dispersively. So what about after 2 rounds of replication? Well, what they saw here were 2 discrete bands, one in the middle of the gradient and one at the top of the gradient, not a smear. So again, the conclusion there is that the DNA, represented in the E. coli chromosome, the E. coli genome, is replicating semi-conservatively. So again, a very simple, straightforward experiment with a very elegant design that answers a very fundamental question about the nature of how the genetic material actually replicates in this organism. Okay, so just like I said with the discovery of DNA as the carrier of genetic information, in previous lectures, once you demonstrate that in one organism, you answer the question in one organism, you have to show that it's universal. So you have to do similar experiments in other organisms. I mean, just because DNA replicates in semi-conservatively in E. coli, it doesn't mean it's universal. So that's what, this experiment was performed and reported, from the mid to late 1950s. So there were other laboratories that were looking at other organisms as well. And ultimately from those experiments, drew the same conclusion. So I want to describe just one of those experiments in a eukaryotic organism instead of a prokaryotic organism. So a little bit more complicated system because, eukaryotes are going to be primarily multicellular organisms as opposed to the unicellular E. coli. So it's the same question. How in a eukaryotic organism, how does the DNA within the chromosomes replicate itself? Conservatively, semi-conservatively, or dispersively? So this is the work of Taylor, Woods, and Hughes. So they used a plant as their model system. So if you're going to be studying or asking questions about DNA replication, where are you going to look? There's different tissue types, different cell types in the multicellular plant. And so where are you going to look? So you want to look at a tissue in which there's lots of mitotic activity, lots of cell division occurring, because then before a mitotic event can occur, you have to replicate the chromosomes and the DNA within the chromosomes. So they chose the root tips. There's lots of mitotic activity at root tips, and I'm sure most of you are aware of that. When you look at pre-prepared slides to visualize different stages of mitosis in a biology lab or a genetics lab, that's one of the materials you almost always look at. Okay. So what did they do? So again, they had the same issue, is that they had to, in order to distinguish between those three models for replication, for DNA replication, they had to differentially label old DNA or parental DNA and to distinguish it from newly synthesized DNA. Okay. So in this, use a radioactive tag or a radioactive label, tritium, tritiated thymidine more specifically. So again, remember DTTP is one of the building blocks of DNA. And so again, the hydrogen atoms in that material can be rendered radioactive. So that DTTP would be incorporated into newly synthesized DNA and then label it in this radioactive form, which is then detectable by exposure to X-ray film. So basically what they did is they started with, fully, so here's a chromosome in the Vishuyafaba genome. And again, remember that chromosome has a double-stranded DNA molecule that runs from end to end. So again, if the cell that carries that chromosome is going to divide mitotically within the root tips, the first thing that has to happen is the DNA has to replicate itself. And through that process of replication at the chromosomal level, what we know is you generate a metaphase chromosome that consists of two sister chromatids. And each of those sister chromatids has a replicated DNA molecule, replicated from the original, replicated daughter molecules that run from end to end within each of those sister chromatids. Okay. So what they would do is they would dip the root tips in a solution of tritiated thymidine for a short period of time. They'd take it out, wash it off, and then they would cut off the root tips and prepare a root tip squash on a microscope slide. And then they could prepare that root tip squash in such a way that you could then visualize metaphase chromosomes, just like I said, the same way you see these cells in a pre-prepared slides in a biology lab or a genetics lab. And so they could then visualize, the metaphase mitotic chromosomes. And this is what they look like. They look like metaphase chromosomes, two sister chromatids still adhering to each other at the centromeric region. So now what they wanted to do is determine the distribution of radioactivity across those two sister chromatids. So what they would do is you take a liquid film emulsion, you place it on the slide above your root tip squashes, you let it dry, and then you put it away and let it expose for a couple of days. And so that film emulsion, it's just like a piece of X-ray film. After a couple of days of exposure, you develop it, and little tiny silver grains will precipitate within that film emulsion wherever there's radioactivity. So this is what they saw. Here's the schematic of it, but here's an actual autoradiograph of these mitotic metaphase chromosomes. And you can see that there seems to be radioactivity associated all along the length of each of the two sister chromatids. So what does that tell you in terms of, how the DNA within the original chromosome was replicating? You wouldn't have that even distribution of radioactivity if the DNA was replicating conservatively. But you would see that distribution if the DNA was replicating either semi-conservatively or dispersively. So from that one simple observation, they could immediately rule out conservative replication for chromosomal DNA of the chromosomes in the Vishuyafaba genome. So now they had to distinguish between semi-conservative and dispersive. So how did they do that? So they used the same experimental design, but instead they allowed for 2 rounds of replication of DNA. The first round of replication in the presence of the radioactive thymidine, and the second round of replication in the presence of non-radioactive thymidine. And then they would observe the metaphase chromosomes, again, consisting of two sister chromatids. And then they would determine the distribution of radioactivity across those sister chromatids. So again, just to follow this diagram here, so here's the starting point, fully unlabeled DNA, one round of replication in the presence of radioactive thymidine. Okay, so there's your metaphase. But then they would allow the cells to complete mitosis, so to complete the cell division. So these radioactive chromatids would segregate into daughter cells. And each of the DNA molecules that run through those chromosomes are partially labeled with, the newly synthesized DNA is labeled with radioactivity. So then they would allow for a second round of replication in the presence of non-radioactive thymidine. Okay, so the second round is with non-radioactive thymidine. And then they would capture the metaphase chromosomes and then determine the distribution of radioactivity. So here's a close-up. This is what they saw. One of the sister chromatids was uniformly labeled with radioactivity, the other one not. So here's an actual autoradiograph of this. You can see here, and you can see here, one of the two sister chromatids is labeled with radioactivity, one is not. So what do you conclude from that observation? So from that observation, you conclude that the DNA must be replicating semi-conservatively. If it was dispersive, then both sister chromatids would have some radioactivity associated with it. So, but you don't see that. You see only one of the two sister chromatids being radioactive. So the DNA within the chromosomes of Vishya faba must be replicating semi-conservatively. So again, a very simple, elegant experimental design that answers a fundamental question, and it extends the conclusions of Meselson and Stahl, and eventually it's universally accepted that for any organism's genetic material is that the way by which it replicates is semi-conservative replication. I should say is that actually this Taylor, Woods, and Hughes experiment was published a little bit before Meselson and Stahl. So even in this more complicated biological system, the multicellular system and multiple chromosomes, they were actually able to answer this question prior to even Meselson and Stahl. Okay, so once it was understood, again, this is, remember Watson and Crick was in 1953. So this is work that's occurring through the mid to the late 1950s, where it was eventually determined that universally DNA replicates semi-conservatively. Okay, so what that then does is, that's not an end point in terms of understanding, but it actually poses a lot more questions. So now the focus for biologists and biochemists and molecular biologists was to try to determine the specific nature of the replication machinery in the cell that allowed for semi-conservative replication. So first of all, for semi-conservative replication to occur, you have to unwind the DNA. So how does the DNA unwind in the cell? That must be a component of this, this complex replication machinery. What's the nature of the enzyme that actually creates the new DNA off of the single-stranded templates? That obviously is a very important enzyme to understand. It's an enzyme that's, responsible for replicating genetic material. And then lastly, again, if you go back to the Watson-Crick model, again, remember the two strands run anti-parallel to each other. Okay, so again, that actually adds a level of complexity to the overall replication process. Because, during a replication event, you have to replicate both of those single strands of DNA that are running in opposite orientations relative to each other. So really the point I'm trying to make here is that once it was understood that DNA replicates semi-conservatively, well, you want to determine the details of the process. What are the different components of the actual replication machinery? So the first thing you would try to understand, again, is the nature of the enzyme that is responsible for synthesis of the new DNA off of the single-stranded templates. So this is an enzymatic activity that we would call DNA polymerase. So an enzyme, DNA polymerase, it has DNA synthesis activity. So again, this is, the mid-1950s is when there were multiple labs who were trying to purify that particular enzymatic activity and characterize it. That would be a very important discovery. So the individual who was successful here, in collaboration with others as well, was this guy here. This is Arthur Kornberg. And so what he was able to do, again, from E. coli, sort of the workhorse model system at this time for molecular genetics, is that he was able to purify a protein from E. coli that had DNA polymerase activity. And, this is in the 50s, late 50s, this work was considered so significant that he actually received a Nobel Prize for that work in 1959. So very soon thereafter. That's how significant this discovery was, or at least considered at the time. So what he did is he purified the enzyme and then he characterized its properties. And very simply, or we're just going to talk about it at a simple level here, is that what he was able to determine is that in order for that enzyme to create new DNA, to synthesize the new single-stranded DNA molecule, is that it required a template. So again, as expected for semi-conservative replication, required the enzyme. This is sometimes you'll hear this form of DNA polymerase referred to as a Kornberg enzyme, again, recognizing the significance of this discovery. And then lastly, there was also the requirement for, the building blocks. So in other words, what the enzyme would do is it would take the individual nucleotides, read the information in the single-stranded template, and that would then determine the sequence of incorporation of nucleotides into the newly synthesized strand, based on the Watson-Crick base pairing rules. Okay, but more importantly, what he discovered is that those nucleotides, they couldn't be in the monophosphate form, they couldn't be in the diphosphate form, they had to be in the triphosphate form. So all four building blocks, more generally referred to as DNTPs, deoxyribonucleoside 5' triphosphates, collectively represent DATP, DTTP, DGTP, and DCTP. So these are the building blocks that are necessary or that can be utilized by the enzyme to create a new molecule of DNA off of that single-stranded template. Okay, in addition to these three requirements that I've mentioned here, there was another fourth very important requirement. There was a requirement for what we call a primer. Okay, so what does that mean? So in other words, you could load up a test tube, with a buffered solution, throw in a single-stranded molecule of DNA, throw in some DNA polymerase, the Kornberg enzyme, and throw in all four DNTPs, and nothing would happen. What was required is that the template strand had to be partially double-stranded. And you make the single-stranded template partially double-stranded by adding this sequence, this complementary sequence that we describe as a primer. Okay, so again, the primer is held to the template strand through normal Watson-Crick base pairing. So what that then does is, again, remember, the nature of double-stranded DNA and the anti-parallel nature. The template strand, as you see labeled here, is 3' to the left and 5' to the right. So the primer sits on that template strand, 5' to the left and 3' to the right. Okay, so that's a free 3' end. And remember, we talked about, the different chemistry that exists at the 5' and the 3' end of a DNA molecule. So what the enzyme has the ability to do is add nucleotides specifically to the 3' end of that primer, or the 3' end of any growing strand of DNA, and only to the 3' end, never it doesn't have the ability to add nucleotides to the 5' end of the primer. So what you can see is that when this primer is positioned on the template, you have a region of double-strandedness and single-strandedness. So the enzyme can recognize that juncture of double-strandedness and single-strandedness, read the single-stranded template, which tells it which sequence of nucleotides to incorporate into the newly synthesized strand, and then synthesize that new strand of DNA by adding nucleotides one by one to that 3' end. So what that means is that any newly synthesized DNA that is created off of that primer is always growing in one direction only. It's always growing in a 5' to 3' direction. So I emphasize that down here. Synthesis of new DNA by DNA polymerases always occurs in one direction only. It's always 5' to 3'. And what that means is that the enzyme always reads the template strand in the opposite orientation, always 3' to 5'. Okay, so this is a more detailed explanation of how the enzyme works, this Kornberg enzyme. So I should say there's actually another name. Generally we refer to any protein that has this type of enzymatic activity as a DNA polymerase. The first form of DNA polymerase that was ever purified and characterized is the Kornberg enzyme. Another name for that is what we call DNA polymerase 1 from E. coli. So what DNA polymerase 1 will do is it'll catalyze the formation of a phosphodiester bond. Again, remember, that's how you link individual nucleotides together in a molecule of DNA. So the enzyme catalyzes the formation of a 3' to 5' phosphodiester linkage. And it uses the 3' OH of the deoxyribose, the last nucleotide of the primer or newly synthesized strand of DNA. Okay? So where does so if the reaction involves bond formation, where does the energy for that reaction come from? Well, again, remember the incoming subunit is in a triphosphate form, but once it's incorporated into newly synthesized DNA, you only retain one of those phosphates. So two of them are lost. So when you lose those two phosphates, you generate energy that allows for that phosphodiester linkage bond formation. So I've already mentioned this characteristic of the enzyme, the idea that how does it know the sequence of nucleotides to incorporate into that newly synthesized strand? Well, it's reading the nucleotide sequence of the template strand. And again, by following Watson-Crick base pairing rules, the enzyme then knows what sequence of nucleotides to incorporate into the newly synthesized strand. And again, this last characteristic I'll just reiterate is the idea is that because of the nature of bond formation by the enzyme, because it only adds to the 3' end of the primer or newly synthesized strand of DNA, is that synthesis of new DNA is always in a 5' to 3' direction. So we'll look at that. This is what I showed you earlier. Here's the template strand of DNA, again, oriented left to right, 3' to 5'. Here it's been primed by a complementary sequence of DNA held together by normal Watson-Crick base pairs. This is a free 3' end here, that 3' OH of that terminal nucleotide. That can be used to form a phosphodiester linkage. So the enzyme will bring a DNTP into this position here, a DNTP that has base pair complementarity to this nucleotide on the template strand. It'll stabilize that DNTP, and then it'll catalyze bond formation, phosphodiester linkage formation. And so you then grow the new molecule of DNA, the newly synthesized molecule of DNA, by adding nucleotides one by one. Okay, so here's another way of looking at this, maybe zooming in a little bit more closely so we can see the atoms of these nucleotides. So here's the template strand running from top to bottom, 3' to 5'. Here's the primer held together or held to the template strand through normal Watson-Crick type base pairing. And here's the free 3' OH, that terminal nucleotide of the primer. Okay, so what the enzyme has the ability to do is recognize this juncture of single-strandedness and double-strandedness in that prime template. Okay, so it'll bind to that site, and then from the pool of DNTPs that are present in the cell, it'll bring in the DNTP that exhibits base complementarity to this nucleotide on the template strand that is unpaired. So then it stabilizes this incoming nucleotide through normal Watson-Crick type base pairing, C with G. So once it's properly positioned and stabilized, the enzyme can catalyze bond formation. And what it will do is it'll cleave off this diphosphate or this pyrophosphate residue of the incoming NTP, retain that terminal phosphate, and the energy released by breaking that bond allows for this second bond to form, this phosphodiester linkage to form. So that's why they have to be in the triphosphate form, is to release that energy that then leads to bond formation, the type of bond formation you see in a DNA molecule, 3' to 5' phosphodiester bonds. So then what the enzyme will do is, again, it'll move down the template strand, read the double-stranded, single-stranded juncture, bring an incoming DNTP that exhibits base pair complementarity to this nucleotide on the template strand that is unpaired. So this would be DTTP, stabilized through hydrogen bond formation, catalyze bond, break the phosphodiester group off, release that energy that leads to bond formation, adding the second nucleotide to that growing strand of newly synthesized DNA. So again, the point of that is that, as it says down here at the bottom, the sequence of new DNA being synthesized by the DNA polymerase is predetermined by the sequence of nucleotides in the template strand. And that's what leads to faithful replication. That's how this enzyme can faithfully replicate an original double-stranded DNA molecule to make two daughter molecules that are exactly the same in base pair sequence as that original molecule. Okay, so there's one characteristic of this enzyme that I didn't point out when I showed you the slide previously. This is down here. So this is the idea is that, okay, so everybody got really excited with the discovery of a DNA polymerase. As I said earlier, it led to the awarding of a Nobel Prize for this work. However, as it turns out, is that this enzyme is not responsible for replicating E. coli chromosomal or genomic DNA. It has the characteristics of DNA synthesis, but it's not responsible for replicating chromosomal DNA. So what that means is that there must be other forms of DNA polymerase in the E. coli cell that can handle replication of chromosomal DNA. Okay, so why does this one not have that ability? So the problem is, as I said earlier, the biology of E. coli was fairly well understood, making it an important model organism for molecular genetics, in the 40s, 50s, and 60s, and even up to the modern day. But the problem is, or what is known in terms of describing the genome, so there's a single molecule of DNA that represents the entire genome of E. coli. It's circular. It's a circular molecule of DNA, and it's double-stranded, and it adheres to all of the characteristics of Watson-Crick double-stranded helical DNA. But the problem is that it consists of over 4.6 million base pairs of DNA. Okay, so that's a lot of DNA that has to be replicated in a time frame of under 20 minutes. Remember I told you earlier is that under idealized conditions in the laboratory, you can get the E. coli cells to divide every 20 minutes. So that means that the chromosomal DNA has to be able to replicate in under that time. So this enzyme can't do that. You see the rate of incorporation of new nucleotides in the newly synthesized DNA is less than 600 per second. So that's not fast enough to be able to replicate that large circular molecule of DNA in under 20 minutes. So they had to find other forms of DNA polymerase in the E. coli system. And eventually they did. Actually, they found there's at least 5 different forms, and they all perform other functions. So you can think about this a second. You know, there's many situations where you would need DNA polymerase activity. One is for replication of chromosomal DNA prior to a cell division. But when DNA is damaged, you know, there's repair machinery that can correct the damage. And part of that repair machinery involves DNA polymerases. So at the very least, you need DNA polymerases for repair machinery within the cell. So there are other situations where you would need this type of activity other than replicating chromosomal DNA. So eventually they discovered, I'm going to describe 3 different forms, the 3 major forms. You know, 4 and 5 are less significant or, you know, less predominant within the cell. So there's DNA polymerase 1, which we know is the Kornberg enzyme. There's DNA polymerase 2, and there's DNA polymerase 3. So as it turns out, DNA polymerase 3 is the replicating enzyme. You know, it's responsible for doing the bulk of the work during chromosomal DNA replication prior to a cell division. As we'll see, the Kornberg enzyme actually plays a role in that process as well, but it's a much more minor role. But we'll talk about that later. So again, by definition, all 3 of these enzymes are DNA polymerases. So what does that mean? That means they all have the same enzymatic activity, and that activity is 5' to 3' polymerase activity. They can add nucleotides to a new growing strand of DNA by catalyzing a 3' to 5' phosphodiester linkage. So that leads to growth of new DNA in a 5' to 3' direction. All 3 of these DNA polymerases do that same thing. Okay? But actually what they found out is that these enzymes have other enzymatic activities associated with them as well. All 3 of them have, in addition to the 5' to 3' polymerase activity, they also have 3' to 5' exonuclease activity. So what's exonuclease activity? Exonuclease activity is the opposite of polymerase activity. It's actually a DNA degrading activity. It's an activity that will break phosphodiester bonds instead of forming them. So you think, well, that seems counterproductive. Why would you have that activity associated with these DNA polymerases? So it's actually a very important role. It's what we call an error correcting or proofreading activity. So again, DNA polymerases are responsible for replicating DNA molecules, for synthesizing new DNA off of single-stranded templates. And so if mistakes are made, you make an error during that replication process, you're going to change the informational content of the molecule. And you can't do that with genetic material. It leads to mutations. So you want to minimize that as much as you can, so you get faithful, accurate replication occurring. So that's what this, so 3' to 5' exonuclease activity means that these 3 DNA polymerases can degrade DNA specifically in a 3' to 5' direction. And so let me go to this diagram here. What that would mean, if this is a 3' end, the same enzymes that can grow DNA moving from left to right, filling in this gap here, they can also degrade DNA moving from right to left. So remove this nucleotide by breaking that phosphodiester bond, removing this nucleotide by breaking that phosphodiester bond. So the direction of degradation is from the 3' to the 5' direction, 3' to 5' exonuclease activity. Okay, so all three of these DNA polymerases have those two activities associated with them. But what they also found out eventually is that the Kornberg enzyme actually has a third enzymatic activity. It also has 5' to 3' exonuclease activity. So again, what does that mean? That means that the Kornberg enzyme can fill in this gap through polymerase activity, 5' to 3' polymerase activity. It can degrade this primer from a 3' to from a 3' to a 5' direction. It can also degrade the primer moving in a 5' to 3' direction. So it can do a lot of things. Okay, and it's not immediately obvious. You know, we know that the 3' to 5' exonuclease activity is proofreading. So when the DNA polymerase makes a mistake, it can quickly recognize that mistake, cut it out, and replace the wrong nucleotide with now the correct nucleotide. So that's the error correcting activity. But why would the Kornberg enzyme have 5' to 3' exonuclease activity? So we'll talk about that in a later discussion. Okay, so this is just a table that summarizes some of these characteristics that I've pointed out. You know, when you compare DNA polymerase 1, 2, and 3 from E. coli, so again, they all have the proofreading activity. They all have the polymerase activity. That's by definition what they do. And then you can see here is that the Kornberg enzyme has this additional 5' to 3' exonuclease activity. But there's one other thing I want to, the other thing that I've mentioned is that, you know, the Kornberg enzyme synthesizes DNA very slowly, 600 nucleotides incorporated per second. But over here, you can see the DNA polymerase 3 has a much higher rate of incorporation. It can synthesize DNA much more rapidly. Okay, so that's a fundamental distinction between the two types. The other thing I wanted to point out, and then we'll wrap up this discussion for now, is the idea is that the Kornberg enzyme, DNA polymerase 1, consists of a single protein, a single polypeptide, within which there are 3 catalytic domains that have these 3 different enzymatic activities. So a single protein, a single polypeptide that has those 3 different enzymatic domains within, you know, that protein. But the DNA polymerase 3, the actual replicating enzyme, has a much more complex organization. It consists of at least 10 different protein subunits, all of which aggregate into this large complex that then has the ability to replicate chromosomal DNA very quickly and very efficiently. So again, a fundamental distinction between the two. So let's look at the Kornberg enzyme in terms of its organization. So this represents the protein, you know, the DNA polymerase 1 itself. So again, this is just a string of amino acids with an N terminus and a C terminus. So what you can actually do is, in what they do, you know, for the laboratory applications of this enzyme, is you can actually break this single protein into 2 pieces. Okay, you know, so through digestion of the protein, such that you create these 2 shorter products. Okay, so, you know, we have the green and the pink. So this is the short fragment or the small fragment. This is the large fragment. So this small fragment, consisting of about 323 amino acids, has a sequence of amino acids within it that serve as the domain for the enzymatic activity of degradation in a 5' to 3' direction. Okay, so that's that 5' to 3' exonuclease activity is associated with this smaller fragment of the original enzyme. The other 2 catalytic domains are contained within the large fragment. So this larger fragment is sometimes referred to as a Klenau fragment. If you've ever worked with the Klenau fragment as a form of DNA polymerase, you know, if you've done some laboratory research as part of your undergraduate career here, that Klenau fragment has two catalytic domains within it, one of which, again, a subsection of amino acids that have the ability to, catalyze DNA polymerization in the 5' to 3' direction, or alternatively, another domain that has the proofreading activity, 3' to 5' exonuclease activity. Okay, so the reason I point all of this out is, again, to just emphasize the fact that the DNA polymerase 1 is a single protein that has these three different catalytic activities. So it's a very simple organization. And it's fundamentally different from the DNA polymerase 3, which is the main DNA polymerase used for replicating chromosomal DNA. And this is what its structure looks like. So as I said, it consists of at least 10 different subunits. These over here and these over here. And you can see the organization is such that it's a dimeric enzyme. So this upper portion of it is, it's like a mirror image of each other. So each of the halves of this upper portion of the molecule consist, I'm just going to point out here, this is the alpha subunit, epsilon, and theta. And you see the same thing over here. And then at the bottom of those complexes, you have a fourth subunit, a fourth protein that we call, beta subunit, or the beta clamp. Okay. And then down here at the bottom, you have a complex of subunits, of which, there's no mirror image. It's just one complex of these subunits. So how does the enzyme actually work? Well, this complex at the bottom of the molecule allows for binding to double-stranded DNA. So if this enzyme is going to be involved in replication, it has to find its substrate before it can do anything. It has to find a double-stranded DNA molecule of, chromosomal DNA within the cell. So it can find that DNA through this binding activity of this complex down here. Once it binds, what will happen is that the two single strands will be separated from each other, or in the double-stranded molecule, the two single strands will be denatured or separated from each other. And each single strand will be fed through this beta clamp to the alpha subunit here and here. So in other words, the enzyme, being dimeric in its organization, has the ability to read two template strands at a time, making it more efficient in replication. Again, remember the Kornberg enzyme only has one catalytic site for DNA polymerization. It's found down here within this large fragment. So the Klenau fragment or the DNA polymerase 1 can only read a single template strand of DNA at a time, just for new DNA synthesis. Okay, so once the beta clamp feeds those template strands, they feed the template strand to this alpha subunit. It's the alpha subunit that has the 5' to 3' polymerase activity. And that's true here. There's two alpha subunits. So you can synthesize off of two template strands simultaneously. In addition to that, you have this epsilon subunit associated with the alpha subunit, and it's that epsilon subunit that has the error correcting activity, the 3' to 5' exonuclease. And then this theta subunit, again, it's unclear as to what its exact role or function is during a replication process, but that may contribute to the 5' to 3'. I'm sorry, the actual role of that theta subunit is unclear at this time. Okay, so this is just, again, a summary of these multiple subunits that are found within that complex that we refer to as DNA polymerase 3, the form of DNA polymerase in E. coli that is responsible for chromosomal DNA replication. And then, again, this is just assigning different functions to those different subunits. Okay, so given that, now that we understand that DNA replicates this is late 50s, we know that DNA replicates semi-conservatively. We know that that process of replication is driven by a DNA polymerase that has been, purified and characterized. So now the question is what are the other components of the replication machinery? Again, before a DNA polymerase can replicate a DNA molecule, you have to be able to denature the DNA molecule, right? The DNA is double-stranded normally. So there must be some way that within the cell, with part of the replication machinery, you can degrade double-stranded DNA, convert it from a double-stranded form to a single-stranded form. This also involves unwinding. Remember, it's a helical molecule. The double-stranded DNA molecule is helical. So that introduces problems during the replication process. If you unwind the DNA, making it single-stranded from its original double-stranded helical form, what happens to the helix? What effect does that have on replication? It has a significant effect. The other question is well, where does again, remember these polymerases, these DNA polymerases require a primer. Where does the primer come from? What's the nature of the primer? And once the primers are laid down and the DNA polymerases start to do their work, how do you remove the primers when they're no longer needed? Another problem associated with replication, again, as I mentioned earlier, is that the two strands run anti-parallel to each other. But now what we've seen is that the main replicating enzyme, the DNA polymerase 3, is dimeric. So it actually reads both of those template strands simultaneously during a replication event. But the two strands are running anti-parallel to each other, meaning that the direction of new DNA synthesis is occurring in opposite directions off of each of those two template strands. So that's a problem that arises during chromosomal DNA replication. Okay, so what we've described again is the basic approach or the basic mechanism for replication, semi-conservative replication. We have a little bit of an understanding of the key enzyme involved in the process of replication, the DNA polymerase itself. But what about these other components of the replication machinery? So this is something that we'll talk about in our next lecture.